When most people think of venomous animals, they tend to focus on reptiles and arachnids, but in reality, venomous creatures can be found throughout the animal kingdom. One group of venomous creatures is the genus Conus, commonly called cone snails. Conus is the largest genus of marine invertebrates, and of all the gastropod genuses, it diversified the most rapidly. Like most mollusks, cone snails have a specialized organ in their mouths called a radula, covered in tiny serrated teeth called denticles. This organ is typically used to both break down and move food for ingestion. In cone snails, a highly modified denticle called a harpoon is used to inject venom into the prey. The harpoon is transported through the snail's proboscis in order to assure accuracy. Most cone snail venoms come in the form of proteins called conotoxins or conopeptides. More than 75,000 conotoxins have been identified. Most of these toxins target various receptors and ion channels in the cell membrane. These receptors and channels are used in intercellular communication, as well as the provision of chemicals needed for cellular processes. There are some conopeptides that individually target calcium, sodium, and potassium channels. All three of these ions are involved in the processes of muscular contractions and neuron communication, among other processes. Reactions to conopeptides can be categorized into three groups: lightning strike cable, where the victim's nervous system is hyperexcited; motor cable, where the neuromuscular transmission is blocked, causing flaccid paralysis; and nirvana cable, where the victims become slow, dazed, or disoriented. Some Conus species have been shown to use weaponized insulin, these snails being the first animals discovered to do so. The snails expel the insulin, which is more similar to fish insulin than their own, into the water for the fish to intake through their gills. In the 129 cases of human envenomation by snails of the genus Conus, 36 victims died. 60 of the known cases of humans being stung by cone snails involved C. geographus, and of these, 31 were fatal. In the 15 cases where the interval of time between C. geographus envenomation and death was known, a third of the victims died in less than an hour. Death is usually due to heart failure and asphyxiation caused by respiratory paralysis. While there is currently no antivenom for any Conus species, survival of victims can be better ensured with advanced life support. Conotoxins are valuable in pharmaceutical research due to their ability to target specific cell receptors and ion channels. Researchers have been able to use conopeptides to distinguish closely related autoimmune diseases, as well as to differentiate between inherited and acquired forms of similar cancers. About 80 or more drug patents have been made that hail from conotoxins. The pharmaceutical and medical potentials of cone snail and other animal venoms are still being revealed. Who knows where they may lead?